We're talking about cell-mediated immunity. We'll get to the humoral immunity pretty quick here, but one thing I want to add to the T helper topic here, remember, they contain a surface marker called CD4s, and cytotoxic T cells contain surface markers called CD8. And helper cells are only going to respond or activate or be challenged by an antigen that is delivered to them by an APC. And the antigen has to be on MHC2. Now, an APC has both MHC1 and MHC2, correct? Because all cells, except for red blood cells, have MHC1. The activation, however, I want to make sure that you realize that it is a two-step process. And so in this two-step process, we need presentation, plus co-stimulation. And this is actually for all of our lymphocytes. So presentation, MHC2, foreign antigen to the T helper cell. And the T helper cell will have some things that will go on to where it will actually then release co-stimulatory enzymes, which will then allow for two things to happen. Cloning. And cloning, when they're fully activated, they will clone. So they clone and they will create two divisions. Most of them are active members that are going to go out and look for other T and B cells to help. That's why they're called helper cells. They're going to go out and look for other T and B cells to help to give them this always happens. One of these days I'll figure it out. To give them co-stimulatory enzymes because it's a two-step activation process. Liken this to a car. Okay, you want to go somewhere. What do? You get in the car, you put the key in, and you turn the car on. That's step one. If you want to go someplace, you have to do more than that. Step two is putting it into gear. So you have to understand that full activation is a two-step process. And when they are fully activated, they will clone and then do their job. And T helper cells' job is to help. But clones always produce a whole big group of cells that are doing their job, and then another group, not so large, of cells that are called memory cells. So whether we are a T cell or a B cell, because we're going to talk about B cells too, it doesn't matter. When they clone, they create two subsets. Big subset that's actively doing their job, and another subset that's called memory cells. So keep that in mind. That's a complete activation. Cytotoxic T cells, they have the surface marker CD8. Again, two-step process for them. And we're going to take a look at the picture down below and go over the T helper cell and the cytotoxic T cell also. So here is our MHC2 on our APC. So APCs always have MHC2. And remember, T cell receptors or TCRs, those are T cell receptors, are going to interact with the MHC2 class on that helper cell. Then that helper cell will clone and make a whole bunch of helper cells that are going to actively go out and help other cells. Plus the clone always has a memory group. That's a smaller subset. The memory group, we're going to talk about this, is no longer naive. Because our little, does it talk about this? This first cell right here is naive. When you are naive, what does that mean to you when you hear the word that you're naive? You don't know something because you're inexperienced. So that's what naive means. This is a cell that has been trained but has never been, has actually experienced what it was trained for. So that's an important thing. Naive, they have gone to school, just like you guys, and you know what you're supposed to do, but you've never done it before because you've never had to actually do it. You've never had the encounter before. So that's an important thing. That's the activation. So these active cells down here of this clone, they are no longer naive. They are now seeking out other T cells and B cells that they can help. That's why they're called helper cells. And they help because what they do, their big job in helping is to co-stimulate. So that co-stimulating enzyme is important to actually complete the activation process. So the activation is 2 steps: encounter and co-stimulation. Key in on and into gear. And we'll always give you 2 groups: active cells in the cloning process and memory cells, which now have actually seen had the encounter, but they have been told you're in reserves. You are the reserves. You only come if we need you again. So these guys are in reserve. Let's take a look at the cartoon here for the cytotoxic T cell. What's important here, it says naive cytotoxic T lymphocyte. Remember, it has CD8 as its surface marker. It still has a T cell receptor that recognizes or will respond to that specific foreign antigen. And here we have another APC at this time. Other cells can do this, but APCs are actually best to do this. And APCs do have class 1 on them. The thing about cytotoxic T cells, they have to have presentation to them via a class 1 MHC receptor. So that's what's really cool about APCs. They have 1 and 2 with them, and 2 goes ahead and works with the helper cells, and 1 is really great, can go in and activate cytotoxic T cells. So APCs have a dual role: activate helper and can activate cytotoxic T cells. But the T cell, remember, has just had the presentation with MHC1. And so now we need second step here, which is at interleukin 2. That's not important. What I want you to know is that there is a co-stimulatory enzyme that finishes the activation process. So helper cell right here is now helping cytotoxic T cell to complete the activation process. And then again, we get cloning. And cloning will give you the worker bees and the reserves. So the memory cells are your cells in reserve. And so cytotoxic T cells are now looking for any cell that has that foreign antigen in its MHC1. And let's take a look at how it does the killing. Okay. So the cytotoxic T cell will bind tightly to that target cell's A prime or apostrophe. That's what I'm kind of using that to make sure that you keep in mind this is definitely a foreign antigen. Because now we have self-antigens, right? We have our own antigens that are self-antigens, and to reiterate, this is a foreign antigen. I put that little apostrophe by it. The binding, so it's hand-to-hand combat to me. It's now releasing the perforin granzyme complex. So perforin, remember, is an enzyme that's going to polymerize and create a big hole in that cell. And then what's nice about this, the T cell leaves and does the same thing to another cell that has the very same problem. When the T cell leaves, we get that polymerization of the perforin, that pore, and then the granzymes. It's a kind of a 2-packet step even here for this process. Granzymes enter the pore. They activate enzymes inside the cell that causes cell death in 2 hours via apoptosis. And so this perforin granzyme complex is deposited by cytotoxic T cells that is designed to destroy that cell that it bound to. So this is hand-to-hand combat. The interesting thing is we do know that cytotoxic T cells can also release this stuff that most people call TNF, It's called tumor necrosis factor. We still don't know the mechanism by which it works, but it is obviously designed for cancerous cells. Let's go in and add to our notes for naive cell. Any lymphocyte who has not been fully activated, or we can say has seen war. Cells that are naive are T or B cells, T or B lymphocytes, helper or cytotoxic cells. And remember, these guys, they have been programmed and if you want to say it in a more technical term, immunocompetent. They have been trained, remember, in the bone marrow for B cells and in the thymus for T cells. So they have been programmed, but no actual encounter. And we can sometimes use the word challenge. When you are fully activated, can do your job, which is co-stimulatory for helper cells for cloning purposes, or actually hand-to-hand combat for our cytotoxic T cells. Memory cells are always produced during cloning for future fights. They are like the guys in reserves. Similar to what the military is. And you only get memory cells if you've been activated. So activation is a major process to actually give you memory cells. Hence, that's another reason why we give vaccines, because we are trying to create a whole line of memory cells for the individual. When you have memory cells, you can respond faster and more efficiently. To another challenge. Memory cells, too, are always activated. They're always ready. They know what to do. The other thing, though, in order for a memory cell to fully do what it needs to do, which is what a naive cell needed to do. In order for a memory cell to actually respond to another challenge, what must it do to be actually responding to that challenge? It must clone, right? So a memory cell must also clone. You have to understand activation is always a two-step process, and you need cloning to make a larger military in order to meet the challenge. But the nice thing is you have a lot of already satellite clinics out there, so to speak, ready to actually encounter the pathogen and then respond to that pathogen by cloning and then directly attacking. When they clone, though, in order for them to clone, they always need the helper cell. Well, you already have also memory helper cells with cytotoxic T cells. And so the helper cell is there already to actually give co-stimulatory enzymes so that they can actually then create a large army of fighters for you. So this is an important thing, because I do want to talk to you too about, you get an infection, that you've had before, and so we get all these memory cells that are activated, cloning, and doing their job for you, whether they're cytotoxic T cells or T helper cells. And T helper cells are helping the cytotoxic T cells to do what they need to do. You guys are following me on this kind of this weird cycle? But this is very important for everything to actually occur. Where do these memory cells go? Where do these cytotoxic memory cells and helper memory cells go? Because if you have a whole bunch of memory, and I told you this system is systemic, where do they go? Yes! It is that simple. They're lymphocytes. And what did we talk about before this chapter? We talked about lymphoid organs and lymphoid tissue. So yes, memory cells, whether they're T cells or B cells, whether they're cytotoxic or helper cells, they are a whole bunch of them, and they don't stay in one spot because that's not going to work if you want to have a systemic response, correct? So these guys will go out and they seed lymphoid organs. They'll go to your lymph nodes, they'll go to your tonsils, they'll go to your spleen, they'll go, they won't go to your thymus, but they'll go all to all these other lymphoid organs because you want to have an active, engaged surveillance system now, correct? That's what this is all about. This is now total surveillance. Again, this is why vaccines are actually so very helpful for this. Treg cells, we aren't going to say too much, but you can actually realize we need to suppress our immune system because once the battle's been won and you've got all these guys ready to fight, you need to tell them, it's time to go to sleep. We're done. Go away. Stop fighting. War's been won. And no more cloning, no more activation type of process. Yes?
Are naive cells only naive if they've never seen a pathogen, or are they naive with every new pathogen?
Naive means they've never seen it. They've never cloned. So that's what naive means. They've been trained. It's just like you guys. You guys go to school and you've been trained, and you'll get into the nursing program and you'll have all kinds of training and stuff about how to insert a catheter. But you haven't done it yet. And so you are naive until you've actually inserted the catheter, whether it's an IV catheter or urinary catheter. You are actually naive until you've actually had the true encounter. And that's what this is too. These guys have been trained, become immunocompetent, but they've never had to really do the job. And so that's why memory cells are not naive anymore. And naive cells are slow. The very first time you have to catheterize somebody, whether it's IV or urinary, you're not going to be like you are after 10 times, right? So naive cells, the whole process of actually mounting the response takes time, the very first time. But once you've had the encounter, once you have been activated, whether you're a memory cell or active fighting cell, you are fast. And so that's why we want to actually have memory cells. Memory cells make us actually really rapid response system. When you are naive and never had the encounter, it's not a rapid response system. It's like, well, I think I should do it this way, or I can't remember exactly if I'm supposed to do this first or this first, and you have to consult with your friends and stuff along that line. And so the naive cell really is good, but is why you end up having memory cells, but they aren't fast. Let's talk about B cells now, the humoral immune response. B cells make antibodies. T cells really require direct contact. B cells, once they're activated, no more direct contact is required. They will make antibodies which go into our bloodstream or our interstitial fluid because they are plasma proteins. And we talked about plasma proteins can actually be part of the exudate for you. Antigen challenge usually occurs in the spleen or the lymph node. Spleen if it's the pathogens in the blood, lymph node if the pathogen is in the soft tissues. You guys follow that? Interstitial fluid, gets in the soft tissues and then gets into the lymphatics and then goes into the lymph node. When we activate our B cells, the initial thing that will happen is cloning, and they call that clonal selection, but cloning happens first, and then antibody production happens second. So let's take a look at the activation process. It's still a two-step process for proper activation. So what did we have to do in our T cells to start proper activation? Encounter or exposure, right? And then what else? Co-stimulation, right? So the two-step process here. The B cell receptor looks very different than our T cell receptors. The B cell receptor always looks like a Y because that's actually a very specific type of antibody on their surface. So here's our B cell receptor, the Ys, and here is an antigen. And what's really nice about the B cells, because B cells also act like APCs, antigen presenting cells. So here it is, here's the antigen floating around in the system. It lands on the B cell that's designed. These are, one of the things that you have to realize, these are designer drugs. This is what they're using when they're talking about monoclonal antibody techniques. These are truly designer drugs. They are designed for only this specific type of thing, nothing else. So here's that antigen floating around in your bloodstream. It finds all kinds of B cells and all kinds of B cell receptors, and they go, I am not designed to recognize you. And until it finds the B cell or the B cell finds it, there is no response. So it's specific for that antigen. Now it binds, as you see here, we have it binding, and we can have also cross-binding occur. But when this antigen finally finds, and this B cell finally finds this antigen and binds, then that receptor actually endocytoses, goes inside the cell to turn the B cell on. So this receptor goes into the cell, and then take a look here at our B cell. Now our B cell is becoming an APC because it endocytosed it, and if it's becoming an APC, that antigen is not going to be presented to the T helper cell unless it is mounted onto an MHC2. So the pathogen which was floating around out externally is now brought inside and loaded onto the MHC2 receptor. The MHC2 receptor will now interact with, if you notice here it says activated helper T lymphocyte. So that means this helper cell has already seen this pathogen and has become activated. It's very important for you guys to realize this helper cell is only going to help out the B cell that the helper cell and B cell have both seen. This is all the monoclonal aspect of things. So now this helper cell is doing the second part of the activation of the B cell process, presentation and co-stimulation. And the helper cell will then co-stimulate that B cell to then do the next thing, which is always clone. Presentation, co-stimulation. Presentation, co-stimulation gives you cloning, and you clone into, well, there it says memory, but let's go ahead and also clone here into activated B cells, which are called plasma cells. If you have a plasma cell, that means that is an activated B cell, and it is producing antibodies. You also have clones of memory cells. So if you notice, the cloning always produces two subsets, a large group that's doing their job and another group that will come back and are basically in reserves and will come back and do their job another day with the next exposure. Our notes here say antigen binds to specific receptor on the B cell. Absolutely. It's very specific for that antigen. B cell endocytoses that receptor antigen complex. And then in order for us to complete our activation, we have to find a T helper cell that knows what to do. So the T helper cell will help the incompletely activated B cell finish the activation process. And when it does this because co-stimulatory enzymes help to finish the activation process. Our fully activated B cells will make plasma cells that make antibodies and will make a subset of memory cells that are designed to remember the infection for their future. So those guys are in reserves. Let's talk about primary and secondary immune response, this immunological memory story. Primary immune response means this is the first time that we've ever seen that antigen. So first exposure to the antigen. We are in primary and secondary immune response, what we are wanting is we are wanting AB production. Whether it's primary or secondary immune response, because antibody production is exceedingly helpful. And remember, AB is antibody. Okay, so what happens? In primary immune response, first exposure takes 3 to 6 days before antibody production begins. Then peak antibody production takes 10 to 14 days. So going from naive to fully activated, it'll be 3 to 6 days, depending upon the individual, before any antibodies are actually being produced and released. And these antibodies are real important. We're going to talk about that in just a moment. Then it takes 10 to 14 days for peak antibody production to occur. So a little bit in 3 to 6, and we'll get our peak production in 10 to 14 days. This is why we like vaccines, because if you get the influenza virus before you get the vaccine, what that means is that you are sicker than a dog for 3 to 6 days. And then you start to feel like maybe you're going to live, and then you know you're on the road to recovery in 10 to 14 days, and you're feeling like you're getting back to normal. So it's like antibiotics. The reason you take antibiotics is you want to get them into your system, and you have to get enough of them into your system to start to feel better, correct? Same thing with antibodies. You have to get enough antibody level in your system in order for you to feel like you're actually surviving the entire process of a bacterial infection. So that's primary immune response for antibody production. Our secondary immune response is any subsequent exposure, and what cells are responsible for secondary immune response? Memory cells. Absolutely. So we are dependent upon memory cells. And remember, the memory cells, those lymphocytes have, whether they're T or B lymphocytes, they have gone all over your body and they have seeded all of your lymphoid organs, because these are a whole bunch of people that don't need to be working right now other than to finding places to live for the next encounter. So this is a memory cell story. Okay, so we will get, interestingly here, peak antibody production in 2 to 3 days. Due to systemic placement of memory cells. In various lymphoid organs. What's important for you to realize with memory cells, they have already been, they have gone to school and they've actually had the encounter, as far as they're concerned, with the pathogen. They've just never had to finally fully release, if they're a B cell, release their antibodies. So we have all these memory cells all over the place. The pathogen gets there. It's basically going to say, reminds the memory cells. This is what you were designed to do. And the memory cells that are helper cells and B lymphocytes will interact because they already have this encounter and are already they're idling by. And so co-stimulatory enzymes hit those memory cells, and memory cells then, in order for memory cells to really work, what do you know they're going to have to do? Clone. And what do you think, if you have a whole bunch of memory cells all over the place, how many cells are you going to have now compared to the primary immune response? A whole bunch more. So first off, you have a whole bunch more cells out there right away, and if you have a whole bunch more cells producing antibody, what's your peak antibody level going to, even though the peak antibody level is 2 to 3 days, what's it going to look like compared to the primary immune response peak antibody level? Really way up there. So secondary immune response is the one that makes you feel better the next day. So that's why I'm kind of tease the people that say, oh, I'm getting a cold, so I'm going to go home and take echinacea. And then they come back and say, it worked. And I go, okay, because I know it worked. But they feel better, and I don't want to have this long, lengthy discussion about why they're actually better. So when you start to feel like you're getting sick, and then the next day you're going, oh, I guess it was nothing, you have had an entire battle going on inside your system to get rid of that, to make it nothing for you. That's secondary immune response. That's why we are such big advocates for vaccinations. So now let's go into that. Active and passive humoral immunity. Active immunity. This means that when you have active immunity, you will produce your own antibodies. Naturally acquired, well, that means you catch the cold, you get the flu, you get a rhinovirus, whatever, you get that, and your mom takes you to some kid that's got chickenpox in the neighborhood, makes you play with them for a while. That's what we used to do before chickenpox vaccine came out. And parents did this because kids getting chickenpox is very darn inconvenient financially, isn't it? If you have 2 working parents and the kids get chickenpox, what does that mean? Somebody's staying home, or they're hoping that grandma's close by, that type of stuff. And in addition to that, chickenpox can cause a lot of scarring, can make you actually susceptible to shingles later on in life. And if you're an adult and you get chickenpox, you can get pneumonitis from it. You can get all kinds of nasty things from chickenpox if you get it at the wrong time in your life. But that's naturally acquired. You will make antibodies to that, and when you get exposure to it again, what will happen? Nothing. Okay. Artificially acquired. That's where we inject dead or attenuated. Attenuated means weakened, can't really hurt you, pathogens or their parts into you, and we call this immunization or vaccine. And, you can try and talk to anti-vacciners about the reasons why they should get a vaccine. Keep it simple, because people who don't agree with you, the more complicated you get in your discussion, the less they listen to you, correct? So just call it an early warning system. I think you'll get a better response with it. What do you mean an early warning system? Well, your system has actually been introduced to this so that it can mount a background military response to it, and so the next time this comes into your child's system or area, it puts the kibosh on it right away. They still won't believe you, but you can do your best. Okay, so if active immunity means you produce your own antibody, what does passive immunity mean? It means you don't produce your own antibodies, but you have antibodies. So where do these antibodies come from? Outside sources. So let's talk about passive immunity for you. Let's talk about snake bites. Let's talk about botulism. Let's talk about rabies. Why are these things so bad? They'll kill you before you can mount an appropriate immune response. So you can have pathogens introduced into your system that will kill you before you can respond to it. So therefore, we need to help you to respond to it if you actually have this encounter. If somebody gets a rattlesnake bite, what are they supposed to do? Just lay there and die? They go to the hospital, the emergency room, and hopefully bring the snake with them for identification. But if it's a rattlesnake, you say, it's a rattlesnake, you know. It had the little rattle, rattle noise of the tail. Unfortunately, we don't have too much of that in our area, but there are other areas that have rattlesnakes and people get rattlesnake bites. So you go to the ER and hospital for what reason? Why are you there? Rattlesnake anti-venom. That's right. So we'll talk about rattlesnake anti-venom. Rattlesnake anti-venom. Where does it come from? Anybody have any idea? Pardon?
Is it from the venom of rattlesnake?
Well, this anti-venom is an antibody against rattlesnake venom. Okay, so you guys have to realize an antibody is against a very specific thing. So anti-venom is an antibody against rattlesnake venom. Okay. We'll talk about how these antibodies actually work on rattlesnake venom in just a moment, but it's going to go ahead and help us eradicate the rattlesnake venom from our system. So it's antibodies against very specific rattlesnake venom. Where do we get these antibodies? An animal. That's right, rattlesnake anti-venom. And the animals we like to use are big animals, and they're called horses. We get a horse, and guess what we do to the horse? We inject rattlesnake venom into them. We don't kill them. we want antibodies from them. Okay. So rattlesnake anti-venom comes from horses. Inject horse with rattlesnake venom. What happens to the horse? It doesn't die, otherwise we wouldn't be doing this. So the horse lives, and the only reason the horse lives is because it made antibodies to the venom, therefore we can call it anti-venom. And how long will it take for us to get peak antibody levels from this horse? 2 weeks, okay. Well, and so do you want to harvest the anti-venom from the horse then, or do you want to do another thing? They'll give you a bigger bang for the buck. Inject the anti-venom again. So indeed, you will have a horse that has had 2 exposures to the anti-venom. The second time you give them the anti-venom, when do you want to harvest? Within 2 days after the injection. And so then you will go ahead, just like they do plasma banks and stuff like that, because where is the venom, or the anti-venom, excuse me, the antibodies? Where is it located? In their plasma, because antibodies are plasma proteins, okay? And so then you'll harvest from their plasma the antibodies to the venom, and then you'll get a nice large batch of anti-venom, correct? So times 2, we inject the horse with anti-venom times 2, and very specific time frames. You want the first injection to have at least 2 weeks before you give the second injection, correct? Because you want to make sure that you have the proper immune response from that. So then harvest antibodies. and then you inject anti-venom into the person with a bite. And so when they go into the emergency room, they get that shot right away, because we want to get that into them right away, because this is so toxic that we cannot mount an appropriate immune response to respond to it. So do you guys understand why the reason you are here is because of your immune system? Now, your immune system has kept you from succumbing to some of these nasty things in your environment. And what you have, your parents gave you, and their parents gave them, and their parents gave them, and their parents gave them. And the ones that didn't have the appropriate response to the exposure died off. Do you see the evolutionary selection of how you are actually here? Because your ancestors were able to actually recognize and fight all the nasties that they encountered in their world, and they gave you that survival mechanism. So passive immunity means you don't make your own antibodies, somebody else gives it to you. And there's two ways to actually have passive immunity. We can inject anti-venom or antibodies into you that we harvested from another thing, or you can ask your mother for help. If you ask your mother for help, you are not a human being walking around. You are a human being in utero. And that's what's really nice about moms. Moms protect their young, and they start protecting their young even before they're born. When mom gets exposed to all the nasties in the world, guess what she makes? Antibodies. And floating in around her system is all these myriad of antibodies. Okay, all kinds of antibodies are already floating around in your system. And if you're a mom, those antibodies are also floating around in your system, and they have the unique ability to actually cross the placenta. So they can actually cross the placenta, and then that means that the antibodies mom made are now in the unborn child. And that's really great, because when babies are born, what kind of world do they enter? A nasty world. But they have already been protected by mom. Mom has infused in them all the antibodies that to the pathogens that she has been encountering in her world, so that the baby can come out with a completely naive immune system, but they have all these antibodies already in their system floating around taking care of things. And then if mom breastfeeds, guess what else happens? Antibodies are also in breast milk. And so mom is continuing to give her newborn child a whole bunch of protection against the nasties out there in the world. So immune system is really, really important. Let's take a look at the antibodies. We call them immunoglobulins. Again, didn't I tell you they're plasma proteins? And from this name, globulin, that should ring a bell, that's a protein, because proteins have fibrous form or globular form, and they have a function based upon one structural and the other is something that does something. So immunoglobulins. We're going to take a look at the basic structure of the antibody monomer. Remember the word monomer. Monomer means building block, correct? And so the antibody monomer is always Y-shaped. So that's why I had some of those Ys that I've drawn throughout this topic. Ys are antibodies, and they're an antibody monomer. There are 4 polypeptide chains held together by covalent bonds. So is that a primary protein structure, secondary protein structure, tertiary, or quaternary? That's a quaternary. You guys, stuff that you actually are continuing to bring forward with you in your memory banks is really cool. Okay, there are 2 light chains, which means they're short, and 2 heavy chains, which means they're long. And the light chains are associated with the prongs of the Y, so to speak, and then the heavy chains are the entire length of the Y, and we've got 2 of them. The interesting thing is the chains, whether they're light or heavy, have an area that's called variable and an area that's called constant. The variable region is the antibody binding sites, which are at the ends of the Y. I said antibody, I should say antigen, excuse me. They're at the ends of the Y. So how many antigens can a Y hold, 2. That's why they're called bivalent. The FC, or the stem region, down in this area, is very important because it's involved in the classical activation of complement. So we've talked about complement before, and the classical activation. We'll touch on it again in just a moment. Now, a monomer is a building block, and it's bivalent, so 2 antigen binding sites. And what's interesting, we have different classes of immunoglobulins that are based on the constant region of the immunoglobulin. So we have different antibody classes, but when you take a look at a class of antibodies, their constant region is all the same. That's how we define their class. Let's take a look at the 5 different antibody classes that we have. We have immunoglobulin. Ig is the shorthand word for immunoglobulin D. It's a monomer, and it's important in activating B cells. So it's shaped like a Y. Important in activating B cells because it sits on the B cell surface. So these are B cell receptors. IgD is the B cell receptor. IgM is a pentamer. Penta is 5. And so if we were to draw this, we would do 1, 2, 3, 4, 5, and then add our little. It is the first class of immunoglobulins released after activation of a B cell. Is it easier to produce a pentamer, or is it easier to produce a monomer? A monomer. This also contributes to a delay in your response for antibody levels. So first class released in your first activated cell. IgG is also a monomer, so it's the lovely Y-shaped guy, and it is the main antibody. This is the one that we want for primary and secondary response. So we get IgM in a primary response, but we also get IgG, and the one we want, the one that does the best job for us, is IgG, and this one can cross the placenta. And this is the one we use for passive immunity. And so a gamma globulin shot is IgG. And so whenever we are giving people antibodies from other animals, we are actually preferentially giving them IgG class antibodies. We'll stop there because it's time to quit. I am going to make a supplemental lecture for the remaining of this chapter because I want us to keep us on a certain timeline so that we can get done earlier at the end of the semester instead of later. So you guys will have a full week to actually study for exam 5 and exam 6. So I want to keep us on a certain timeline. So it will be listed as supplement for this weekend. Please make sure you review it because on it will be information about RH and blood typing problems and erythroblastosis fetalis, which is RH incompatibility with the newborn.